population, we identify that variant actually that's responsible for the major, when we identify genes, we identify few lines actually from QTL mapping, and then further we in progress, we either develop a backcross population to narrow down further the region where the QTL is located, right, sir?
Well, someone was asking, can we say mutational breeding is old school breeding? No. Breeding is still the old school. We're just trying to minimize the number of cycles or number of resources used, be it QTL mapping or association mapping or any other technique. We right away replace the traditional breeding, be it phenotypic selection or mutational breeding. We're just trying to minimize the time and the resources needed.
Yeah, artificial mutations can be employed to mutate QTLs, yeah. But all we need to know is why are we doing that? The objective is more important.
All right, it's 10 minutes past our schedule time. So I'll just start with the presentation. Good morning, everyone. Today, I want to talk about association mapping. I'll start with a lecture followed by a demo in Tassel. And for the demo, it's going to be a 2-day demo because we have multiple things that we have to do. In over years, QTL analysis and linkage mapping have been streamlined and we can have everything in a single software. But for association mapping, we have few other steps beyond what the softwares can do. That's basically for a better interpretation and a better idea of the results. So we'll split our demo today and tomorrow, and we'll try to interpret some decent results out. And the data set we have been using is something I have analyzed already in Barley. So I have modified the marker names and the sample names. And if you so that we don't have any further issues. And before I go with that, just want to start with the GitHub page. We have uploaded the QTL analysis workshop, the data used for QGene, and at the same time, I have uploaded the genotype and the phenotype that we use for Tassel demo today. By the end of the day, we will be uploading the Day-4 presentation too, and the YouTube link should also be ready, and Abraham should provide you with the link through email. So with no further ado, I just want to start it. Other than linkage mapping and QTL mapping, we will come across a few more concepts or a few more heads. So don't panic with all the terminology. Unless you work with this, you might feel something new. But as I said, even though there are many new topics that we would cover today, I'll try to keep it simple. No need to panic. And once you start working with it, everything will look easier and simple. So just a revision of marker-assisted selection. We described earlier before QTL analysis that we need at the end of the day, we need marker-assisted selection to save on the resources used by the breeders and also to decrease the hybrid development time. So basically, this is a simpler method compared to phenotypic screening, and even in the seedling stage, without growing the plant and looking for the phenotype, we can carry out selection in the seedling stage. So we'll be saving a season completely. And here single plants can be selected with high reliability, and we can accelerate the line development or the hybrid development in breeding programs. In general, we can at least 2, but possibly 3 or 4 backcross generations could be saved using markers. If you're talking about annual crops, you're talking about at least 4 seasons or 4 years based on how you grow it. But if you're talking about a perennial, it's a lot. For example, a mango or something, if you could save 4 cycles, that's definitely something right. And on the top of it, this marker assisted selection is high throughput in the sense if you have the right resources, you can screen 10 million plants in a day. But with phenotypic screening, you can't even screen 1,000 plants in a day. A single person cannot do that in the field. So that's the reason marker assisted selection is of high throughput. And this makes pyramiding of genes easier, meaning you can select for multiple QTLs or multiple regions easily. And basically, this could be achieved by identification of markers or QTL or genes associated with the trait. And the 2 common approaches are QTL mapping and association mapping. QTL mapping, we have already discussed about it, and we'll talk about the association mapping today. Just to follow up the same slide that we have it earlier, if you could see, if we could screen the F2 seedling stage, in F3, you could see the number of plants we grow compared to that traditional pedigree method is 1/4 of it. That saves us a lot of time and resources. And association mapping, basically this is also called as linkage disequilibrium mapping. And it's a method for mapping QTLs that take advantage of linkage disequilibrium to find associations between phenotypes to genotypes. Basically, what it is saying is, this is a method where in your DNA, you have a gene or a QTL that is responsible for a trait. If you have markers close by, or technically, if you have markers that are in LD or linkage disequilibrium with that QTL, we could identify those QTL regions and we could use those markers for marker-assisted selection. We'll talk about linkage disequilibrium about a little bit because that forms the sole core of this association mapping. So basically, LD refers to non-random association between 2 markers or 2 genes and QTL. Here is an example. Basically, we're saying we have 6 individuals. This is not a biparental cross. These are 6 random individuals. In terms of crops, it could be 6 plants or 6 varieties of plants. And if you see here at locus 1, you have a nucleotide A and 3 individuals and nucleotide T in the other 3 individuals. Similarly, at locus 2, you have G and C. These 2 are said to be correlated in simple statistical sense, or here we call it as non-random association. And here the R² value, which is a measure of linkage disequilibrium, which we'll talk in a minute, is 1, or basically the correlation between these 2 is 1. If you take a second scenario, it's the same, but these 2 markers are associated with trait variation. In this particular example, you can see dark brown and light brown. So you could say, after all the analysis, these 2 markers or these 2 locus are associated with this trait variation. This linkage disequilibrium can occur between side locus that are on the same chromosome, that are distance apart, and even on different chromosomes. So this is a big difference from QTL mapping. In QTL mapping, we're only talking about on the same chromosome within 50 centimorgans, that things. But here, LD could exist between markers between regions on different chromosomes or even on the same chromosome which are distant apart. So how do you measure this LD? There are basically 2 measures. One is the D prime and the other is R². D prime basically only considers the recombinational history, and if D prime is not equivalent to 0, we call it as they are in linkage disequilibrium or LD. On the other hand, we have R square that considers both recombinational and mutational history, and and this in simple sense lies between 0 and 1, and R square of 1 means they are in complete linkage disequilibrium. With my menus, I'm not able to see everything out. So and here R square of 1 means they are in complete linkage disequilibrium. In simple statistical sense, R square is nothing but the squared correlation value. As you know, correlation exists between -1 to 1. So if you square it, the value lies between 0 and 1. So here are a few examples to show how we calculate D prime and R square. I just don't want to go with the calculations, but to explain, if you could see at locus 1, you have a red allele and a blue allele, and green allele is associated with yellow allele. This is a perfect correlation, or a R square of 1, meaning they are in perfect LD. In another scenario, your red one at locus 1 is associated with both blue and yellow, whereas green is associated with the yellow, and here your R square is bit low, 0.33. And this is the mutational history, which I don't want to go in detail. In third case, you have both red and green alleles at locus 1 associated with both blue and yellow at locus 2. So this is a case where R square is 0, D prime is 0, meaning they're not in LD or they're not correlated. For the calculation purpose, we might use another measure called as partial LD. Basically, since I said the LD is nothing but the square correlation, we could use some covariates when calculating a correlation coefficient. And when we square that value, we call it as a partial LD. So basically, we're including covariates. Covariates are nothing but other factors that are not directly responsible, but that affect the trait. For example, if you take a plant population, the country might be a covariate. If you're talking about humans, age might be a covariate, or the sex of the human might be a covariate. So those are the covariates. So partial correlation is necessary because unlinked loci can be in LD simply because of population structure and kinship. Meaning like when you have population structure, which I'll talk in a minute, and kinship, it basically leads to, it says they're correlated, but when you use them as a covariate, you will end up saying they're not that much correlated. So this particular value is called as the partial LD. That's really good if you're trying to pinpoint the genes. Instead of using a complete LD, using a partial LD helps us identify genes close to the markers, or minimizing from say 100 genes to 10 genes. Other important visual representation or to get an idea of LD is LD decay and LD heat plots. Basically, LD decay plots are the distance over which LD persists will determine the number and density of markers and experimental design needed to perform an association analysis. So once you have all the marker data, we develop an LD decay plot. And here, if you have the LD decay, I'll show an example. If you have decay that could extend to mega base pairs, then a few markers are sufficient in theory. But if you have a short LD, you need multiple or thousands or millions of markers. As an example, let's assume I have a 100 MB genome and my LD is 1 megabase pair. By theory, all I need is 100 markers or maybe 200 markers, such that we have one marker in each megabase pair. But if your LD is only for say 1 kilo base pair, you're talking about 1,000 or million mark, but 1,000 into 1,000 about 100,000 or million markers. So we need more markers if the LD is short. So here is an example. On the x-axis, I have the distance in mega base pair. This is for soybean, using 2 populations of soybean and around 35,000 markers. And I used a nonlinear regression approach. And if you could see, if I take a value of 0.5, this corresponds to a correlation of 0.7 or more, or minus 0.7 or less. So if I take that, my LDDK is approximately 2 megabase pairs. 2 megabase pairs. Since soybean is 1,000 megabase pair, in theory, 500 markers or a bit more than that is sufficient, assuming that the LD stays same throughout the genome, which is not the case. So since I have 35,000 markers, I'm theoretically saying I'm well above the necessary range, and that is a really good thing. One other previously, when we started association mapping, this is using 1500 markers in 130 lines, and this is using a centimorgan distance. And as you could see, just a second, I'm missing my mouse. Okay. As you could see, if I take the same 0.5 value, I'm talking about approximately 2 centimorgans distance. The main properties of LD based on that graph you could see is with the distance, LD decreases. And if there is a random mating between that samples, the LD decreases by 1/2 of the generation. Meaning if you have say 100 samples that you collected marker information, but for your phenotype, if you're doing the next generation of it, you're losing half of that LD. So we should be careful with that we get the phenotype and the genotype information from the same generations. And as the number of generations increases, LD is limited to small distances because you have more rounds of recombination and more rounds of random mating, LD will be decreased. So just as an example, if you have land races wild types, which have been existing from a long time, you have limited LD or LD blocks are small. But if you have cultivars that are developed in the last, say, 30 years or 50 years, you have long LD because it's only 50 years, whereas for land races and wild types, it could be some 10,000 or 5,000 years. The other thing, a visual representation of LD that helps identify the candidate genes are called as LD heat maps. Basically, these are also called as disequilibrium matrices. As I said, we calculate LD as an R² value, which is the squared correlation value. And we'll be using that value to find the linear relation between different markers. Here as an example, I found a QTL region on chromosome 7 from 6 to 7 megabase pairs, out of which, and these are all the markers present. If you could see, these are the markers that are highly significant that I found. There are 2 known genes that are responsible for this particular trait, iron deficiency chlorosis, that leads to yellow leaves in soybean. As you could see, they're in good LD. The red, orange, yellow colors are reasonable LDs. The light blue colors are, or the green colors are very low LD. So I have red areas corresponding to these markers, meaning these markers are able to explain the variation of this gene because they are in LD. So I have been talking about linkage disequilibrium or LD a lot, and we need to know what factors are increasing or decreasing LD so that helps us identify the population to use for association mapping, and at the same time, our expectations, what could we expect from our results. The first and the foremost factor that increases LD is the mating system. In mating system, if it is selfing, it reduces opportunities for effective recombination. So meaning, if you have a selfing species, the LD would be higher. But whereas if it's a cross-pollinated one, definitely your LD will be smaller. Domestication. I know most of you know about domestication. About 10,000 years ago, plants were selected for specific traits. That's how we ended from tea ocean to maize or wild beans to the regular day beans, wild tomatoes, the smallest wild green tomato to red, juicy, sweet tomato. These are all part of domestication. And we have many other crops that have been domesticated over the years, including rice and wheat. So in terms of genetics, what happens is you have an ancestral population. So where the allele frequencies, see here you have 8 samples, out of which 2 samples have a different allele. And here 4 samples have a different allele. During this bottleneck or the domestication process, you are selecting only this particular combination. So you end up increasing the allele frequency of it. So that is increasing your correlation, which in turn is increasing your LD. So your mid selfing and domestication result in increased LD. The other factors that contribute to increased LD include genetic drift. Basically, during genetic drift, you lose rare allelic combinations. So you will be left with, say, initially, let's look at the same example here. Here, the rare allelic combination is, color a bit. So this is a rare allelic combination present only once. During genetic drift, there are chances you lose these allelic combinations. So that again increases your correlation LD. When you have gene flow between populations of the same species, it induces new individuals or gametes, and so you have more combinations, so that leads to increase in LD again. And in terms of selection, positive selection, it's more like domestication where you select for desired traits. So genetically, you're selecting for desired allelic combinations. On the other hand, if you have population structure, meaning if your samples could be distinguished into different groups, either based on geography or genetics, then we call it as a population structure. That also increases LD by creating different allele frequencies. So on the other hand, what could be the factors decreasing LD? One thing is recombination. As mentioned earlier, one generation of random mating leads to half decrease in LD decay. So recombination basically decreases your LD because due to crossover, your allelic combinations gets changed. And the second thing that is the population. If you have a larger genetic variation, there is a high chance there will be faster LD decay because of historical recombination. I mean to say, if you have a very large population, in that particular population, you have individuals acting differently or different amounts of recombination that leads to breakdown of allelic combinations that in turn leads to decrease in LD. For example, in corn, in land races, you have until 1 kilo base pair, whereas in diverse inbred lines, which have been developed in the last 50 to 70 years, you have 2 kilo base pairs. And you have recent 30-40 years of elite inbred lines where LD could extend several hundred kilo base pair because you're intentionally selecting for some traits which in turn are selected due to certain A combinations. And you have random mutations that could decrease or change your alleles that would lead to a decreased correlation or a decreased LD. Gene conversions, which is a bit rare. So with all these things, with all these factors influencing LD, should we grow with the increased or decreased level of LD? First of all, both are useful for one purpose or the other. If you have narrow genetic diversity and long extent of LD, they are good for coarse mapping and you need few markers. And if you have genetically diverse populations, short LD-DK, they're good for fine mapping and you need more markers. Okay, let me explain that. Let's take, for example, sorghum. Sorghum is grown in India and Africa a lot, and you have environmental variation. You have, and at the same time, it's grown in US too. You have different environments, and it has been a part of the domestication process, and it has been a wild crop for years. So there, you have a larger genetic diversity. So when I include samples from, say, US, samples from Africa, samples from India of that particular sorghum, you're talking about shorter LDDK because you don't have the same allelic combinations throughout. So they are good for fine mapping. At the same time, we need many markers, few millions of markers to pinpoint a few traits. On the other hand, let's take a crop that's grown only in India, say, for example, recently jute. Jute is mostly grown in Bangladesh and northern India. So there, the genetic diversity is too narrow, and there are not many breeders working on that. So there's a very narrow genetic diversity, and it has been established only a few years ago. So you have long LD blocks or longer LD decay. Or the other says you have allelic combinations that have been existing from a long time. So when you have such populations, you end up coarse mapping, meaning your marker might be a megabase pair away from the QTL or the gene of interest. And in these cases, you need fewer markers. So based on your crop, based on the samples, based on the populations and everything, you can decide whether to go with fine mapping, coarse mapping. So SNPs, do you want to go with chips or GBS or next generation sequencing or a few SSRs? It all depends upon your species and the population. So coming to, since we're done with the basic concept of LD, let's look at the main concept of association mapping. Basically, you have two kinds. One is the genome-wide association mapping. The other is the candidate gene association mapping. For example, using other prior literature or some QTL analysis, we know that the QTL is present on, say, chromosome 2 from 10 to 20 megabase pairs. We could just use the markers within that region on a different population to narrow down the candidate gene. In those cases, we call it as candidate gene association mapping, but that is rarely used because finding markers within them and manually doing is harder than doing a genome-wide association mapping where you can use all the markers and use SNP chips or other systems. So if you look at the literature or if you look at all the scientific talks, you might be hearing the association mapping or GWAS a lot. Why is that? Because there are a few advantages of association mapping. First thing is you don't need a recombinant, you don't need a segregating population. And you know the time it takes to develop a segregating population, not just the time, the resources. As an example, if you want to develop a recombinant inbred line population, you need approximately 8 to 10 years or even more based on the crops and other things. And at the same time, even an F2, you're talking about 2 years. And at the same time, the resources, growing them in the field or greenhouse, crossing them, making accurate crosses and all those things. And at the same time, association mapping, we don't need such things. We can just randomly take a few samples out and work with it. I will show you a few examples on the kinds of populations we use in association mapping. And the other main advantage is higher resolution. Here is an example or a research showing that with association mapping, you can see that you can have 100 base pair resolution approximately, meaning you could find a marker close to the QTL or the gene of interest within 100 base pairs. Whereas with other things, it could be a mega base pair or even more. And at the same time, the time taken to association mapping is approximately 1-2 years compared to other process which takes 5 or 10 years based on the time based on the species and how many generations you have per year. So when I talk about resolution, here is a good example showing the resolution. So here you have a double hybrid mapping population, RILS And this is the diverse germplasm. Here I have A and B or biparental crosses where we did QTL analysis. And if you could see there about F9 generation for this real populations. You could see here the value is approximately 10 centimorgans to identify a QTL, whereas in RILs, you have until 5 centimorgans because you have more generations, more recombinants, so there is a higher chance of getting a bit lower QTL resolution. But whereas with association mapping, your resolution could be as less as 1 centimorgan. It again depends upon the population used and the markers, but theoretically, you get a very low resolution or a very close marker. So here is a typical example of an association mapping. Let's take a berry number in grapes as an example. And here you have different berry numbers. Let's look at if you have 3 SNPs. This is the high LDSNP where you have C and T. C for mostly greater than 10 and T greater or less than 10. And the other case, you have T greater than 10 and A for less than 10 berry size. So T, and other case, you have both G and A spread throughout. So if you look at the means of each, so for example, this particular marker, if you look at the means, G mean is 10.8 and A mean is 10.2, well, there is no significant difference. So this is not associated with berry number. On the other hand, if you have T and A, 13 and 8 are the means. So there is a significant difference between both the means, and you could see that particular marker is associated. In this particular example, they have been saying is it's not genotype, but that's the functional SNP. And you have another marker close by this particular marker, which kind of resembles this pattern, and you have a difference of 12.4 and 8.6. So you have a p-value of 0.037. So in theory, compared to this A, this B marker is associated with your functional SNP that is responsible for this trait variation. So I have been saying there are a couple of problems with association mapping, and we have solutions too. One is population structure. Let's say, example, let's assume we have 2 subpopulation. In subpopulation 1, you have one SNP T most of the times, and in other you have G most of the case. Due to environmental variation or other factors, you have height generally greater than 10 here, and for subpopulation 2, you have less than 10. And you have disease resistance mostly susceptible for subpopulation 1 and mostly tolerant for subpopulation 2. If I do not include subpopulation as a covariate, I might end up saying this T is associated with susceptibility and greater height, and G is associated with a tolerant and small height. But that's not the case. We have to control this for subpopulation because each subpopulation is rich in its own alleles. The other thing is kinship or family relatedness. Even though you have a diverse population, you might have samples with similar parentage or grandparentage, or over a few generations, you might be having the same relatedness with the family. With that, what happens is you have same LD blocks spread throughout the genome. Maybe 50% of them shared, 40% shared. It depends upon how deep the crosses are. And even a perfect outcrossing species to there is no perfect outcrossing and self too, right? You have other things like mutations or some things happening that could carry over. So all these things, the relatedness between these individuals also affect it. And we use a kinship matrix and use that as a covariate in our analysis. So how does this differ from QTL mapping? First and the foremost, in QTL mapping uses biparental populations, and here we use natural variation. And in QTL mapping, you have limited recombination events. Whereas in association mapping, since this is a random population, each with a different parentage, each with a different lineage, you have many recombination events. With QTL mapping, you have low resolution and the QTL only covers a few centimorgans. Whereas with association mapping, if you use the right amount of markers, you can get close to even 10, 100 base pairs of the gene or the marker responsible. And these are useful for discovering rare alleles, and whereas association mapping are for common alleles. With association mapping, we cannot identify rare alleles. So as a comparison, linkage is correlated inheritance of loci, and that is through physical connection on a chromosome. They both the markers or the QTL or the gene should be on the same chromosome within a certain distance. Whereas LD refers to correlation between alleles in a population and it's non-random. It could be, they could be distant apart on the same chromosome or even between different chromosomes. In QTL mapping, LD is generated by mating design, whereas in association mapping, it's a reflection of the germplasm collection. And in QTL analysis, in mapping populations, LD is influenced only by recombination in the absence of segregation distortion. Whereas in association mapping, LD is influenced by selection, mutation, mating system, population structure, and many other things that we studied in the previous 15 minutes. So, given that GWAS is completely dependent on LD, we looked at a few factors responsible for increase in LD and decrease in LD. So, for the species under study, based on, say, domestication history or many other, the gene flow system and other things, we can design the association mapping experiment and how many plants, how many replicates, what should be the marker system, what should be the depth, and what is the resolution expected, all those things. So let's briefly look the GWAS methodology. The first thing is identifying a decent diverse population and phenotyping it for the trait of interest. And then you genotype to get the marker information. And then you have imputation, where because any marker system, you end up having some missing values, both in phenotype and the genotype. Imputation is a phenomenon where you try to get those estimates. And then, just like linkage map, we clean them based on certain minor allele frequency, meaning the alternate allele should be present at a particular frequency. As an example, if you have 100 individuals, one individual has A allele and 99 individuals have B allele. There is a high chance you will not find any mean difference between those alleles, and their minor allele frequency is only 1 out of 100 or 1%. We remove those kind of markers. And if the population is large, 1% is fine, but by default, we use a 5% allele frequency. And then we use population structure and kinship as covariates to control or to avoid false positives. And then we run a regression with multiple models. I'll explain in a bit. And then we do model testing to identify the best model. And once we identify the best model, we identify significant markers that could be used for marker selection. Then we do a stepwise regression to narrow down the QTL regions, and then we identify candidate genes. This is in general the association mapping experiment, but until the publication stage. But if you really want to use that in your further work, you need to validate them in other populations, because you don't want to identify the same markers. You don't want to identify markers in every experiment you have. If you could have a couple of markers or 5 markers that are significant in multiple populations, that makes your work a lot easier. I'll explain a few examples and in the next few slides, so that should help you understand better. So let's look one by one. When I talk about population, some of the examples are population with overall diversity. For example, land races that are throughout the world. And you have regional populations with local diversity. For example, cultivars that are grown in one state. In my case, I said North Dakota. So when you take regional population, your LD blocks will be high, fewer number markers are okay, and you end up coarse mapping or your QTL and the marker are bit apart. Whereas if you take land races throughout the world, it has history of a few hundred or thousand years. You have smaller LD blocks, and the resolution could be smaller, or you could identify markers really close to the gene or the QTL. So here are a few things. Germplasm bank and elite material. Germplasm bank are nothing but the core collections of all the land races and the wild types, whereas elite material is something the cultivars and the inbred lines that are traditionally grown in our state or close by our area. Main things to note are the level of LD. Here it's low and high because of the years it took to generate those things. Elite materials, which it's only like 20 to 50 years, whereas germplasm bank, if you include all the land races, it's few hundred to 1,000 years. And here the population structure is medium and it is high. The reason it is high here is if you are a plant breeder, you know you never work with a single created one. You might be working with say rice blast, you might be working with some insect resistance, you might be working with some number of tillers. You might be working with different traits and you select different sets of individuals for different things. So automatically you are selecting different alleles leading to a higher population structure. And in germplasm collection, since they're highly variable, you have a high resolution, meaning you could identify markers or genes which are close to the real QTL or the hypothetical QTL. Whereas with elite lines, this is comparatively less. So the second part of our work is once we identify what population do we use, we phenotype it. Meaning we collect, say, for example, plant height information or some things. And definitely you need replicates. And at the same time, you need data from multiple locations to capture G by E interactions. The reason is we never grow, even though breeders, sometimes for some things we grow plants in the greenhouse. In reality, the varieties are not grown in the greenhouse and you have varying climates over years. So initially, if we could test data from multiple locations or multiple years, that would be really good. So the question comes, how do we analyze this data? First of all, you could analyze the data for individual location. For example, if you take rice from Tamil Nadu, Andhra Pradesh, Karnataka, and if your intention is to release variety for each of these areas differently, then you need to analyze for each individual location. But if you want to do some functional studies, or if you plan to release a variety for all the places together, you can do for combined locations. In either case, if you have multiple replicates, multiple environments, you can use adjusted entry means, also called as statistically called as LSMEANS. So let's assume you have from three different locations. You have data from three different locations, and there is a G by E interaction. So you see a variation in the phenotypic values. When you do LS means or adjusted entry means, it basically adjusts for that environmental variation. And the analysis in using SAS or R or any other softwares is really fast. You're talking about a couple of minutes to 10 minutes hardly. At the same time, you could use location replication as covariates in your final association mapping step. Here, data is multiplied and analysis is slow. Meaning, if you have three locations and if you have 100 individuals, instead of working with 100 individuals, for each location, we work with the same 100 individuals. So it will be combined. We're talking about we will be working with 300 individuals. Instead of combining the phenotype, we have individual phenotypes. But here, your data is multiplied n times and the analysis is comparatively slow. So I talked about adjusted entry means. So here is an example where I had 143 samples. And I had from 3 locations, 4 reps, 2 ratings. 2 ratings is like at the, if I remember correctly, at 30th day after planting and 60th day of planting. So in my case, my intention is to find a marker that could identify iron deficiency chlorosis early in the stages. So for that, and since the 3 locations are about 200 miles apart within North Dakota, I used an adjusted entry means. As a simple example, if I do a simple mean, these are the simple mean values. And if I do an adjusted entry means, by adjusting for G by E interaction, they vary a bit. So overall, this will give you a better estimate of the QTL or the markers. So this is the statistical formula that we use for phenotypic data. Your final Y or your final trait is dependent upon the genotype, the replication, location, intercept, G by E interaction, all these statistical things. Let's not talk in detail about this. The second part of an association mapping experiment is genotyping. So again, based on the crop, based on the resources, whether to go and based on the LD, as we already have an estimate of LD based on the plant type and the species, we could go with low density 200 SSRs or 2000 SSRs, or medium density where you had golden gate assays for 1500 markers or so. And then you have high density chips. For example, this is a bit old. Arabidopsis right now has 500,000 SNPs on Ephemetrix chip. Soybean has 500,000. Wheat has 90,000. Barley has, I think, 70,000. So with Ephemetrix chips, which is on the right side as an example, you can get all the 70,000 or 90,000 data points in roughly for 96 samples in a couple of hours. But it's a bit costly compared to other low or medium density things. And you have next generation sequencing where you have thousands or millions. For example, one of my work 5 years ago, when I did 1x coverage in soybean, I had about 79,000 steps. And when I do 3x coverage per individual in a wild bean pool, I had 10 million steps. And nowadays, one of my sorghum panel, which is funded by Gates Foundation, USDA, and multiple resources here, I work with 1,500 samples of sorghum collected from US, Africa, a few from India. There I have about 65 million SNPs and about 10 million Indias that I'm working with. And I mentioned the thing about imputation. Imputation basically refers to getting those missing values. And now it has been extended to include even the phenotype too, but let's talk about the genotype. Basically, it's predicting missing genotype based on likelihood. It increases the power of association mapping. The earlier softwares, even today, most of the softwares, if you have missing marker data, that marker is excluded from the analysis. For example, I start with 10,000 markers and 5,000 markers have 1 or 2 individuals missing data. Then at the end, we are only working with 5,000 markers. But if I could impute those missing values, I could work with 10,000 markers, leading to an increased power. Here is an example. So you have these markers where they're a bit distance apart. But by imputing, I added 3 more markers here, increasing the resolution. And at the same time, this could facilitate meta-analysis, meaning combining different sets of analysis. And there are multiple softwares that could do that. Likelihood-based, Beagle, FastPhase, Phase, and Bayesian-based or BIM-BAM, things like that. Nowadays, I use Beagle version 5. It can impute a few thousand individuals, in my case, around 1,000 individuals, about 10 million markers on a server by itself for about 2 to 3 days. So to explain better, I have an example that I created. So you have raw data. Here a question mark refers to missing data. Here a question mark refers to missing data. And as you could see, we have missing data for each and every individual and about 4 markers. So what it makes is it makes haplotypes. Haplotypes are nothing but what are similar or close by. If you look here, individual 1 and 3 have similar patterns. So these two make up a single haplotype. And whereas individual 2, 4, and 5 have similar patterns. So these form another haplotypes. So basically the software makes haplotypes initially. Then, okay, since at all other loci, they are 100% similar, this could be a G and this could be a T because 1 and 3 are related. And in this case, again, this could be C, and this could be a C based on other things. So it calculates a likelihood. Again, it keeps changes the values, it's multiple kind of permutations and combinations it works with, and finally gives us a final data set which is better or more similar. When we started this imputation in early 2007 or 2008 time, we had a doubt. How good is the software or how reliable can we think we could use imputation? Because at that time, some of the markers we were doing in our own lab. So if we have missing data, we could always go back and rerun that particular marker again only for those samples. So as an example, we tested soybean. We tested about 12 markers in 100 sample size for the missing data. And after imputation, we found a match of 99.99%. So from then, we started relying on imputation a lot because we feel that exactly captures most of the issues. And this particular estimating likelihood is generally repeated n times and with the different starts, and finally outputs the one with the best likelihood or the highest likelihood. The next part is once you complete the imputation, there could be some sequencing errors, which we expect to be 1%, or some of the models we could expect even 5%. At the same time, so we need to remove markers that are present at a lower frequency. And also for LD calculations that could lead to bias. At the same time, rare variants have little effect to discover QTL. If you know a bit of statistics, if you're looking for ANOVA or t-test, for example, as I said earlier, you have 100 samples, one with A allele, 99 with B allele. There is little chance you could find a mean difference between that 1 allele and 99 alleles. So those are called as rare variants are really present in a smaller population size. We remove them. And if you could see here, with increase in minor allele frequency, the power increases. So as I said, there are a couple of things that we need to control for. Population structure and kinship. So basically we estimate covariates. Basically we estimate covariates and control for population structure. We use different softwares like structure, and the other option is PCA. I'll show you in a minute what they stand for and how they look like. So here is a case control study in humans where you have two populations. Population 1 is mostly with blue alleles and population 2 is mostly with red alleles. This is similar to the example I showed earlier. So if we don't use a population structure, we might lead up to saying that controls have red allele or cases, the blue allele are higher in the cases. So when I talk about structure plot, this is an example from rice. So if you could see, the samples collected from different areas belong to different groups. These particular numbers, I mean, like the African samples all are mostly a single group with 100% or 99% genetic variation from the same group. Where if you see the Middle East, they're a combination of Europe and a little bit of themselves and a little bit of South Central Asia. So here this is a representation of all the samples with their genetic memberships in the populations. And here each color represents a single population, and I think we're looking at 7 different populations. So once we run the software structure or other things are BAPS, admixture, instruct, so on, what happens is you end up saying that individual 1 belongs to subpopulation 1, 90% of it. All the 3 add up to 100% or 1, and the individual 2 belongs to subpopulation 2, individual 3 belongs to subpopulation 3, and so on. So some of these methods assume Hardy-Weinberg equilibrium and time consuming. For example, if you want to run structure on some 50,000 markers and 1,000 population, you're talking about the recent fast structure takes about 2 to 3 hours, whereas the traditional structure takes about a week time on big large servers. And for running structure on any of these, you cannot run all the markers at once, because since you have an LD, you need to remove markers based on LD and select a random subset to make the computational process easier. Well, this all looks a bit complex. If you're studying population genetics, this is one major thing that you have to study. But if you are into mostly association mapping, this is an alternative for that, developed in 2006, principal component analysis. Basically, your data is reduced into smaller number of dimensions, and this summarizes the overall variability among individuals, the divergence between groups, and the variation occurring within groups. So here is an example of common bean, and you could see the first 2 components, and each color represents a different geographical group collected. As you could see, there is variation kind of. This forms a 1 group, this forms a 2nd group, and this forms a 3rd group. I mean, this is just based on 2 axes. If we include multiple axes, we're able to split those populations into groups, or the entire sample set into groups. So the question comes, how many principal components? If you know a bit of statistics, you know if you have a sample size of n, the number of principal components also n. As an example, if I have 200 samples, the number of principal components generated is 200. So how many, should I take all the 200 or just a part of it? And here each principal component explains a certain amount of variation, and it decreases with the increase in number. Meaning the first component explains say 30%, the second one explains 25%, third one explains 20%, so on. It decreases such that the last few principal components are useless, or they have only 0.001 variability or something. So you can use say 25% variation explained by the components. So it could be 3 or 5 or 7, could be a random some number based on your sample set, or some 50% variation explained. Or if you want to use statistically, you have a p-value estimation based on Tracy-Widom distribution, or you have Welliser's minimum average partial test based on Schreiner. And if you know a bit of statistics, there are about 12 to 15 types I know for selecting the number of principal components. So that's beyond this lecture. Or you can take top 3, top 10. It's all based on the variation explained by your sample set. And recently, we have been using stepwise to determine the number of PCs. Meaning, if you use stepwise regression, it takes 1, 2, 3, say, let's start with 10. Using a stepwise regression, it might end up having only 3 that are significantly differentiating the populations. I mean, your trait. So in the end, you might have PC1, PC3, and PC7. And one thing to remember is that based on the sample data, your principal components that explain 25% or 50% is fixed. Unless you use the stepwise regression where you include the phenotypic data too, the rest of the time it's the same number of principal components. So if you have n traits, for example, if you grow in the plants in the field, you're not just collecting one trait data. You just don't collect per plant height and leave it, right? You collect number of tillers, or the tiller length, or the leaf angle. whatnot, any other agronomic character that you think is reasonable is collected. So you could use the same number of principal components for all the traits, unless you use stepwise. And then you have a kinship, which controls for relatedness. So here we use this as a random effect. And this one, it's a matrix of all by all individuals. And this one is used to estimate genetic variation, genetic variance, and residual variance. As you know, population structure describes a remote common ancestry, whereas relatedness refers to recent common ancestry because of common parents. There are two basic types of kinships. One is identity by descent and the other is identity by state. Identity by descent is based on the pedigree information. If you have the pedigree information, like for example, a cultivar X is because of parent A and B after backcrossed with B again at F2 and so on, all the pedigree information. Or you have other coefficients which kind of estimate the identity by descent, but we don't use that mostly unless you're a breeder and you really want to use that. Most of the case we use identity by state. This is basically a similarity matrix. It's the percentage of shared alleles or the distance between two samples. So for example, sample 1 and sample 2 were testing 1000 markers. Sample 1 and sample 2 have 900 markers in common or the same allele. So the value will be 0.9 in the matrix. And there are other ways based on the samples. You might need to separate descent based on similarity. But let's not worry about it. Excuse me. And once you had the phenotype, the marker data, and you estimated the covariance, population structure, and kinship, the next one is running the final linear models or the regression models. Again, this is a bit statistical thing. All we are trying to say is you have two basic kinds, GLM and MLM. GLM is only when you have fixed effects, and MLM is only if you have both fixed and random effects. So your phenotype Y is linked to your marker, your structure, your kinship, and an error term. So let's not worry about all these terms. This is a common thing that you see in papers, so just included that. And here, regression analysis is performed with only one marker at a time. So, and you have the variance estimation, residual variance, and genetic variance calculated for each marker, and we call it as exact tests. They're implemented in SAS, EMA, FAST. These are the different softwares. Also, to save on computational burden, you can estimate the genetic variance and residual variance only once, and we call it as approximate test, and it's called as MIX or P3D algorithm, and it is implemented in a few softwares. So you could see here, as I said, for each marker, we run the regression equation again. So if your kinship, if you estimate a single value and use it for the same thing, it's faster. Whereas if you estimate this K value every time, it's comparatively slow. But with the now modern softwares, things are a lot easier. So other than the regression equation, we could use other tests based on your phenotype. You could use a Wilcoxon test, which is a non-parametric t-test. And here no tests are included, no effects or no covariates are included. And then you have a logistic regression when your phenotype is logical. If in humans, if you look at association mapping, basically there in humans, you cannot create crosses or biparental crosses like plants, right? And at the same time, most of the times your phenotype is screened as presence or absence. Example, cancer present or absent, no relevance to the stage or anything. In that case, logistic regression is one thing. So if you take, for example, cancer presence 1, those are called as the cases, and cancer absent 0, and those group of individuals are called as the controls. And at the same time, you have generalized linear model where your phenotype is discrete, like 1, 2, 3, 4, 5, or small, medium, tall, things like that. Then we use a generalized linear model. And a generalized linear model can include both fixed and random effects. But nowadays, whether you use Gamma or Tassel or Gapit or any other highly developed software, it considers generalized linear models, mixed linear models, all possibilities. So recently, there are other approaches developed. If you're more into statistics, you might understand this better. Even if you don't understand this, just forget about it. In simple things, the basic t-test says your phenotype or your trait is linked with your marker and error. Whereas in GLM, you have a fixed effect like population structure. In MLM, you have both structure and kinship. In MLMM, you include a covariant, one of the marker as a covariant, just like composite interval mapping. And in super, it's a different thing. In farm CPU, blink, these are different options. But right now, most of the time, even today, we use only these two methods, GLM and MLM. So we need to run multiple models. The reason is for each population and phenotype, population structure and kinship may or might not have an effect. So to test that, we test multiple models such that your phenotype is equivalent to just marker and error. And your phenotype has marker, structure, and error, and marker, kinship, error, phenotype, marker, structure, kinship. And as I said, kinship is an IVS matrix that we use once, and structure is mostly a PCA matrix that we use. The number of PCs varies, but for our demo purpose, I'll be using 25%. But recently, for most of my work, I have been using PC1, PC1 as single thing, PC2 as another thing, PC3 as another model, PC4 as another model, until 50% variation. If it is 10 PCs, I'm using all the 10 different models all by itself and with a combination of kinship. And I'm selecting the best model based on 25 models. And here is a model selection criteria. Basically, the idea is the residual errors follow a uniform distribution. So visually, you can look at them, or you could using a QQ plot or using a MST. Which I will show you in Excel how to calculate that, because I was the one who proposed that particular MST methodology for this association mapping or selecting the best model. So this is just a procedure on how to calculate the MST values. Let's not go into the details. Okay, we started with all the data. We ran multiple models. We selected the best model. So among the best model, what markers are significant? It's based on the lowest p-value. So we can take a bond for any correction, which is 0.05 divided by number of markers. Or you can take multiple corrections like FTR and PFTR tests, or you can take permutation tests, bootstrapping tests, family-based tests, and many other things. And if you go through your data, read a few papers related to it, you'll get a better understanding. And finally, let's assume you ended with 10 markers that are significant. There might be 3 markers which are really close to each other, maybe within 100 base pair. And all the 3 are explaining the same effect or the same gene effect. In that case, and as a breeder, you don't want to use 10 markers in your breeding program. So you need to reduce the number of markers to 2 or 3 or 4 to make process easier. For that, we use a method called a stepwise regression. This is one method I proposed in 2010 or 11. So basically, it accounts for LD between the markers and minimizes the number of QTLs or markers for marker-assisted selection. I'll show you an example. And it considers both interchromosomal and intrachromosomal LD. And you don't need covariates because you're including only the significant QTLs, and it gives the combined effect of all markers. Meaning, if you have 10 markers and each explains say 10% phenotypic variation, combined, it's not 100% variation explained because they are correlated, they are close by. The combined effect only could be say 25% or 35%. Using a stepwise regression, we will be able to identify that. Then yesterday we came up with the word epistasis. Epistasis is, this is again one thing I proposed in 2014 in one of my plus one paper. So let's say you have two markers that are significant that are present in different chromosomes or on the same chromosome. In ANOVA, we just include M1, M2 and an M1, M2 interaction effect. So if they're significant at a certain percentage, we say there is epistasis or they are related even though they're different chromosomes. For example, with one of my study, I had 33 significant markers, and out of which 528 two-way combinations or interactions were tested, and only 5 interactions were significant at p-value of 0.001. So if I look at the functional scale, though there are 2 genes in a pathway where A gene leads to B. So obviously the markers linked with A and marker links with the gene B are interacted. And the next comes the variation explained, or the tassel gives you an output, but there is one more appropriate method or a decent method where you could explain the variation explained by each of the marker, where you have a null model and a full model, I mean reduced model and a full model. In the full model, you include marker with other factors. In the reduced model, you take the marker out and the difference between these two, not a clear difference, but this one could give you the R square likelihood ratio. And the next thing is effect of allele. So you have parent 1 allele and parent 2 allele, or a reference allele and a variant allele based on the sample set and you're using. You don't have parents here. So between alleles, if I change an allele from T to G, what is the effect? To calculate that, let's say your reference allele mean is 10, variant allele mean is 16. So that half of the difference, so we call the additive effect as 3, which is the difference between these two is 6. 6 divided by 2 is 3, and that is the additive effect of the alternate allele. Okay, the next step would be we identified significant markers. As a breeder or for a marker assisted selection, we need to generate some possible combinations of them. Let's assume we have 3 significant markers, and for an SNP, they are biallelic, so only 2 things possible. So there are 8 possible combinations. And let's assume these are the means of each of those combinations for our sample sets. As you could see, let's assume this to be a disease score. This is the combination I would be using to select it. And this is the combination I would completely avoid. This is the combination I would completely avoid, so on. And to understand the functional aspects, we can annotate the SNPs using softwares like SNPFF, where they say whether they are synonymous, non-synonymous, they are present in the gene or in the intron, in the exon, intergenic region, in the transcription factor, or whatever. And for the QTLs, I generally consider the markers that are significant in stepwise regression and that have a partial LD with the adjacent marker to be a QTL block. So I have a marker. I try to calculate LD or partial LD with adjacent markers on either side. Excuse me. And the region where the LD is greater than 0.6 is my LD block or the QTL block. In one of my paper, I combined if LD blocks are within 10 kilobase pairs in soybean, I just included them as a single block. So just to outline, you have phenotype where you calculate means, adjusted means, and you have markers where you calculate imputation, minor allele frequency, and the covariates. And then you multiple models, you select the best model, you identify the significant markers, do a stepwise regression, identify the allele effects, QTL regions, and do an SNP annotation. That completes association mapping. So once you do association mapping, what's next? You can evaluate these markers on different genetic backgrounds. You can verify them using either using functional assays like nowadays CRISPR-Cas or WIGS or any other silencing knockout things to make sure that the gene we identified in association mapping is associated with the try to understudy. And in terms of breeding, we could use those finally identified 3 or 4 markers for marker-assisted selection. So I would quickly run through some of my projects over the years just to show you that what are the different kinds of populations and the different kinds of marker systems that could be used, just to create an idea where association mapping could be used. One thing is, in cadmium accumulation, we used 96 near inbred lines or nils from 2 field seasons, and we used 9,000 marker Illumina or Infinium chip. And as you could see in both the years, you could see there are 3 distinct populations. In 2006, there were 4 distinct populations. We tested multiple models. We selected the best model that is close to the perfect diagonal. And these are the values. I'll show you how to estimate the lowest value. And we found 2 markers in 2010 that are highly significant with a very good R-square. And for cadmium in the durum wheat or the pasta wheat in US, they accumulate higher cadmium, and cadmium is a heavy metal that's not good for health. So we found 2 markers. In traditional method, identification of cadmium percentage in a seed takes approximately $38 for each sample. And if you're talking about 10 breeding crosses and into 100, you're talking about 1,000 samples, you're talking about $38,000. But if you use the same 2 markers, which is nowadays 2 cents per sample, you're saving a lot of time and at the same time, a lot of money too. So it's like 1,000 plants into 2 cents, $200. And with the DNA extractions, maybe it's like $1,000 or $2,000 more. That's far less than $38,000. And with this, you can get it in a week. With the other cadmium tests, it takes a month or 2. You might lose the season. The other one was germination in barley. We studied 8 breeding programs. Data was collected from 4 years. I mean, the phenotype was collected from 4 years. You had 96 lines each. And we used 4 regression models. If you could see this particular thing, this is the combined all the 32 breeding programs over years. And this particular black line is the best close to the regression line. And this is, we did the Washington, North Dakota, Minnesota, each state separately too. And in the combined block, we found a QTL on chromosome 5 end. But when we look for individual locations, we found different other QTLs. For example, this is from AB, I think it's Canada. We found on 3 and 5, and here we found 1 and 2, and 5 was not that large. Whereas with Montana, we found it on chromosome 5 again, and with North Dakota, we found on 5 again. And here in lupinus, we did plant introductions, 122 of them, and here we used AFLP bands, out of which 892 polymorphic bands. And we found 2 markers with a very good R-square. And this is in wheat fusarium head blight. Here we used BC1F7 lines and BC1F6 lines, which are about the result of multiple crosses. If it is from the same cross, you can't use association mapping. And we used 2,300 dark markers. And we identified QTLs on 2A, 5B2, and 6B. And then we have iron deficiency chlorosis in my PhD, where we did 140 breeding lines and 1536 golden gate assay marker system. And these are the different models we tested. And we found about 9 markers that are significant. And these are the genes, known genes in Arabidopsis or other things that are associated with iron deficiency. And we found them to be close enough. And once we identified those 9 loci in 2005 and 2006 populations, we tried to confirm them in multiple other populations. And we saw these are all advanced breeding lines. And we saw that these 2 markers have been continuously can be used for the advanced generations too, whereas the other markers did not have reasonable R-square or a reasonable p-value. And then the same ideas we did using GBS data with 79,000 data points, but after minor allele frequencies, we had only 35,000 that we used. And we found one on 20 and 1920s. And in 2006, we found one on 7 and 9. And in addition, to confirm that, well, that's not related to association mapping, just to confirm, we took the bulk segregant analysis, 15 resistant types which had IDC range of 1 to 2 and 16 susceptible where the range was 2.3 to 4.7. And we identified SNPs between these two things, and we ended having chromosome 1, 3, and 19 to be significant. And these are a few references if you want to read more about them. And with that, I would be completing the lecture. I know it's a bit of information for all of you, but it is what it is. It's a bit more new concepts. Now let me quickly jump into TASL and show some basic things because we have one more day for learning this association mapping. I'll just show you an outline on how to load data, how to estimate covariates, and we will end there. So stand by with me maybe 10-15 more minutes. So this is TASL, a really good GUI software to estimate or to do association mapping. You have GAPIT in R, you have Gamma in Unix, and many other things that work in Unix environments. I use mostly Gamma, GEMMA, because I can work with say at least 10,000 individuals and 10 million markers in 10-20 minutes. But TASL is a bit slow when you're talking about large data sets. Here, the main things to note are file, open as. Before I go with that, let me show you the marker data, and these two are in the GitHub page. I have two phenotypes. This is one and this is two. This is basically, disease 2 is AUDPC. If you're into plant pathology, you might understand. Area under disease progression curve. So you calculate the area of the leaf on say some day 1, day 30. And after 30 days, you take it again. The difference is AUDPC. The other one is a scoring of disease. And as you could see, we have 136 individuals and 2 traits. On the other hand, I have this marker data for all these samples, around 6,500 markers. And this is the format. If you look at the TASL manual, you will understand this. And here for this, again, it should have the heads like trait. Here it should have the head numeric and marker. So with these input files, TASL, file, open as, make best guess. Instead of we selecting what it is, let it make the best guess. I'll switch off the depths because we're not including depths in our VCF filer and things. Okay. And here you can select multiple files at the same time. I'm selecting both genotype and the phenotype file. Open. If you could see here, your phenotype is numerical values. So it's under numerical. And this is the sequence. Okay. It's not updating, it's a bit slow. Let's look it back in a minute. Okay, and here it's saying there are 3 columns, number of rows is 136, matrix size is 272. Here it should update it. They just released a new version last week and I updated that. I think that might be causing some of these issues. I'll open as in best case. Let me open only the genotype. Okay. It has 136 samples, 6940 markers. Okay. And it should display information here. I'm waiting for it to happen. Okay, let's look for the phenotype first. And if you have missing values for phenotype, you could use options here to impute that phenotypic value. For genotype 2, you can create input files for Tassel, I mean Beagle V4th version. So you can use Beagle further to impute it, but we're not going to do that now. And other than that, you have multiple options that you could play with the data, like joining them that we are interested in. If you are converting them into numerical genotype, if the alleles are ABCDs, so on, you can find a filter by minor allele frequencies and many other things based on our necessity. But for our work, this is the genotype file. I'll go with analysis PCA. Give me a minute, it's not updating for some reason. Analysis, relatedness, PCA. Here you can select either number of components or based on the minimum eigenvalue or the total variance. I'm selecting based on the number of components, and I don't want the eigenvectors. Let me give this a 10 value. Hit OK. Here are my PCs or the principal components, and here are my eigenvalues. If you look here, eigenvalue, the first one explains 10%, second one 5%, third one 5%, fourth one 3%, and these are the cumulative. In our case, let's go with say 25% cumulative variation. So we will need 4 principal components as a covariate. So for that, what I would do is, since I know it's 4, let me save this file. You can save the data as wherever you want to. Okay, and it will save this file. I will delete this, which you can do using file delete data set, and I will recalculate it. I'll delete this too. Select the genotype again, analysis, relatedness, PCA, and I know I need 4 principal components. I don't need eigenvalues. Just hit OK. Just to see how the population structure is this for, you can see do basic graphs using results, chart, and you can select an XY scatter. We're interested in PC1 and PC2. Not much population structure. You cannot clearly distinguish between the groups, right? So this is reasonable. If it is a really higher, then it's better sometimes. It's better to association mapping independently in each of the subpopulations. Now in our case, this is a reasonable estimation and we can go with this. The second thing we want to estimate is the kinship analysis, relatedness, kinship. You can go with centered IB as the default. And here we have only 2 alleles, AB. Sometimes you can have H heterozygous ABH. And if it is SSRs or something else, you can have up to 6 alleles. So that you can have ploidy levels too. Hit OK. I think the updated version has a problem calculating the kinship. So just as a backup, I did estimate it already. Let me show you that. So it has 136 individuals, all samples by all samples, matrix of similarity. So first, the diagonal would always be 1. Since we Tassel, I need to go with a little older version. I'll see, and if I have some information on this, I'll update you guys. But for today, we just I did not show you how we collected the phenotype because that's beyond the scope of this workshop, and the markers, which I said it was based on a chip. And the imputation was already performed because it takes a few days to perform that. So again, that's beyond the scope of this workshop. And in this case, we used a software called as fast phase. If you want to use the advanced Beagle version, it should be a VCF format. You need to have the reference genome with the reference allele and the alternate allele. So coming back to our requirements, we're done with population and phenotyping. Genotyping and marker data is readily available. Imputation was already done. I just calculated PCA 4 PCs and kinship that are necessary to be used as covariates. Tomorrow I will show you how to do the multiple models of regression, both GLM and MLM. And then I will show you how to do model testing. And finally, how to select significant markers, do a stepwise regression, identify QTL regions, and how to identify candidate genes based on the significant markers. And again, validation is beyond the scope of this workshop. So we completed 4 steps. We'll be doing the remaining important 3 steps all tomorrow. With that, I'm open for questions.
Hello, Divya.
Divya, do we have anything?
Yeah.
Can we do association mapping in biparental populations?
Questions, sir.
I think ma'am's internet connection is not stable.
Yeah. Okay.
You hear me?
Can we?
Hello?
Divya, yeah.
Yeah, there are lots of questions here. I think I answered for that question because that's already you explained. Say the biparental populations are most ideal for
Linkage mapping, QTL mapping.
Yeah, linkage mapping and QTL mapping. And there are other questions like how to identify candidate genes after identifying markers associated with the trait in the crop's genomic resources.
We need to know position of the marker. I'll show you an example tomorrow. For example, we identify the marker on chromosome 2 or 10th megabase pair. You can pull all the genes if it is already annotated and whole genome is sequenced. We can pull genes in and around maybe a few kilobase pairs based on LDDK, and within those genes, based on the annotation or previous literature, we could identify the genes, or we will say all the 10 to be candidate genes.
And there's another question, do we use LD values as a criterion to, yeah, that's the one you said, to filter out the markers for association mapping. Yeah, that's what is.
Not for association mapping, the only criteria would be minor allele frequency. LD is just to get an estimate by if we have the sufficient number of markers or not. And at the same time, for candidate gene identification, how much should we go? For example, as I said, if we find a significant marker at 10th megabase pair position, do we want to look 100 kilobase pairs on either side, or 1 megabase pair or 10 megabase pair? It all depends upon the LD we calculate.
Then what is the best way to calculate LDDK? Now, I kind of mentioned like you can go for good softwares because there are many models that you have already explained to calculate
LDDK
LD.
is calculated in our this TASL too. LDDK can be calculated here, but we have programs in SAS and R that we use mostly.
Yes.
But the TASL can handle it too.
Yeah. And can we go association mapping with 15 SSR markers and 300 genotypes?
Yes, definitely. How 55 are they? If they are all on a same chromosome, that will be really good. So that that's a reasonable estimate, I would say, for SSRs. But if they are throughout the genome, and again, it depends upon the number of chromosomes. Are you talking about 5 chromosomes or 50 chromosomes? So it's based on the crop. But by theory, if
That's
it's on
true.
a single chromosome, it's decent enough.
Yeah. And for
What's
100
the
population, how many? Oh, I guess for 100 individuals, how many markers are sufficient?
depends upon what kind of population it is. Are you using cultivars, land races, wild types, worldwide collection, or collection within a state? It's based on the diversity. You can decide on the number of markers. But by theory, the larger the population size, the better it is. The larger the markers, the better it is.
The another one is can we find PCA for 2-year population? I didn't get it. So this is
Mm,
what is a question. Can we
yeah, I mean, I think if we combine data from 2 years, can we find a PCA?
PCA? Yeah.
Well, if you're using different samples, if you have marker data for both of them, definitely we could see if there is variation among the seasons or the years.
The another question is why LD is known as gametic phase equilibrium, disequilibrium. Yeah.
Uh, I don't know why it is called, so it's just called. I don't have a
Yeah.
decent answer for that.
Yeah, it's kind of you can Google it basically.
Yeah.
What are the effects of genetic polymorphisms in a population? I mean, it's a lot of diversity
Yes.
that you
The higher
encounter.
the diversity, the better it is. I mean, association mapping is not possible without that diversity or genetic polymorphism.
Yes. How can we do haplotype analysis?
Haplotype, if it is a few markers, even you could do it in Excel or TextPad. But if you're good at R or some programming, you have multiple programs or you can code yourself. I personally, if it is up to 5 to 10 markers, I use Excel because it's all you need is a concatenation of your 10 markers and calculating the mean in R or even in Excel. And then I could do that.
So, what is the minimum population size for GWAS? Depends on the number of markers and
And the diversity levels.
diversity levels.
But nowadays, if you want a decent publication, if you have a higher diversity population, 100 is fine. But if you're looking for a very low diversity confined to a single region, then maybe 200, 300. Again, also it depends upon the marker systems.
Then, how do we identify whether the markers are significantly associated with the trait of interest? Which alpha level we use? Some use, I mean, significance levels. Some use 0.05, others use 1, 0.5, and 0.05.
That's a very good question. Depends upon the trait. For example, if you're looking for traits like yield, there is a very less chance you'll find a QTL with a very low p-value. In that case, it might be different. Whereas if you take for say disease resistance or some plant height controlled by few genes, few QTL, you will have a lower p-value and a higher R square, assuming that the markers are close to the QTL. So in general, you cannot take 0.01 or 0.05 default. Based on the number of markers, we use FTR, PFTR corrections, or we use Bonferroni corrections. And in my case, I use a different approach called as bootstraps if I have more than some 50,000 or 70,000.
Then what is the p-value that can be used as a cutoff in Manhattan plots if no significant SNPs in the standard cutoff?
Well, Bonferroni is a simple thing to calculate and get top of our head. But again, based on the trait, you can use 1 into 10 power minus 4 or 10 power minus 3 as the cutoff and use that as a cutoff too. But you should be able to support your selection of p-value.
Then how can we select lead SNP or peak SNPs?
Based on the p-value, once you sort based on the p-value, the topmost are your lead SNPs or the peak SNPs. In terms of Manhattan, they are called as helicopters. Because Manhattan is an area in New York where you have the tallest buildings. That's the reason they're called as Manhattan
Manhattan
plots.
plot. I mean, what's the criteria to select peak SNP? Again, from the p-values, right?
Mhm.
That's the same question. That's the same kind of question. Is it possible to use populations above 30% missing value and markers of above 20%?
Yeah, definitely. You need to use a decent imputation programs with a higher burn-ins or higher replications. Because, for example, if you take Beagle version 5, it uses 5 burn-ins, 5 iterations as the default. If you increase them, it increases the accuracy. You could definitely use that.
The graphs in Tassel are not attractive for publication. Any way to make it more attractive? I think R is the best.
R is the best, but that it needs a bit of coding. But the thing is, in R, there is a program called as RCMDR. I'll just type in here. If you could install that as a library, you can get all the R graphs as GUI based, just like Excel click and do, but you get very good decent graphs. It's a library, RCMDR. And someone asked, what's the use of SNP in bioinformatics? Well, SNP's the advantage is, you know the sequence, it's easy to get the positions, first thing. Second thing, you can have thousands or millions of them in your population.
Yeah, I think the questions are, that's it.
Okay. If you have time, please go through the example data set, try to work with the steps so far I have, and there's a YouTube link, and I should have all the slides also posted, so that should be helpful. And someone asked me how to score SSR alleles for association mapping. That's a good question. How many alleles do you have? As said, you can do, if it is an SNP, you can just simply convert into a 0-1 format, or you can convert into 0-1, 2, 3, 4, 5 format. And Tassel, if I remember correctly, can accept until 6 alleles. All right, then. I hope to see you all tomorrow, because so far it's just a lecture in association mapping, a few more concepts, and only a little bit of the analysis work. So tomorrow we'll finally analyze our data and try to get to the end portion of it, identifying the markers, genes, and the QTLs. I'll see you all tomorrow then.
Can I end the meeting, sir?
Yes, Abraham.
Yes. Divya ma'am?
I think I have a poor acoustics here because some people are drilling something at the top. So I'll keep myself muted and we'll see tomorrow then.
Sure, ma'am. I have already provided the YouTube live recording link here, sir. Apart from that, I will be sending the mail.
All right then.
On the same link, they can join by tomorrow, sir. And also, I'll be
Okay.
reminding them.
Okay.
Thank you, students.
All right then. Have a good day.